Welcome back. In continuation to our discussion about breathing and exchange of gases, today we will discuss about transport of gases. So, starting with the transportation as such, the whole content of atmospheric air is not as similar as what is present inside the alveolar cavity. The composition of alveolar cavity air is quite different as compared to the alveolar the air present in the atmosphere. Now, if we start looking and seeing the composition of individual elements present in the gaseous environment outside in the environment, we will see that there are various kind of elements that are there, and there is a clean differentiation or differences between the air present in atmosphere and the air present in our alveolar cavity. So, in the next slide, I will try to discuss about what are the different variation among the gaseous elements present in the alveolar as well as atmospheric content. So, if we start looking in this slide carefully, we will see that the atmospheric air is having quite different percentage of nitrogen, oxygen, carbon dioxide and water. When somebody is inspiring the air inside the alveolar, inside the respiratory passage, it is getting kind of a fortified with kind of water molecules. So, the whole air is humidified. Next is the alveoli content, which is quite different as compared to the atmospheric as well as the humidified air. And then in the last, one can see the nice correlation in the slide about the expired air. So, this is just in nutshell about giving an idea about regarding all the gaseous element present in atmospheric air and in the alveolar cavity. Now, having said that, the differences are there in both atmospheric air and alveolar air. There are certain reasons behind these differences, and some of the reasons behind this is again highlighted in this slide. So, one can nicely see that alveolar air is only partially replaced with the atmospheric air. So, if we have this alveoli, the air coming 100% is not getting transmitted completely into the alveolar side. So, there is a partial exchange of gases from the atmosphere to the alveolar content. Again, there is a constant absorption of the oxygen into the alveolar side, and similarly there is a constant diffusion of carbon dioxide out from the tissue side. So, these are some of the stuff related to the differences between the atmospheric air and our lung volume. And lastly, we have this moisture that is coming from the respiratory passage in the air. So, that is causing a humidification of the whole air cavity, the whole air passage, and that is why the composition of air is quite different as compared to the atmospheric air. Now, if we start looking into the gaseous exchange or the transport of gases. So, we are discussing about the transport of gases. The major part of the exchange is divided into two fractions. So, we will discuss two fractions of this transport, and these are divided in transportation of oxygen and second is transportation of carbon dioxide. So, we will cover these two topics separately. So, we will cover first the transport of oxygen and then we will look into the transport of carbon dioxide. So, first we will start looking into the transport of oxygen. So, the first form is with direct red blood cell, and the second thing is in the form of plasma. So, these two are the distinct form of transportation media. So, either oxygen directly transported in the form of red blood cell or in the red blood cell and then in the plasma. We will discuss the two parts. So, the major part of oxygen transmission from red blood cell is comprising of 97% of the total transport of oxygen, and remaining 3% is from plasma. Now, red blood cell, we will discuss this red blood cell in context of transportation, but we do need to understand why this red blood cell is important and how it is transmitting the oxygen. Now, if we start looking into red blood cell, we will discuss about plasma a little later. So, in red blood cell, if we look in the next slide, one can nicely jot down the actual composition of or actual configuration of a red blood cell. Now, this red blood cell is slightly biconcave in nature. So, these are biconcave structure, approximately 6 to 9 micron in diameter. So, 6 to 9 micron is the size of human red blood cell. This is biconcave in nature, and approximately 34% of total weight of this RBC, in short we will call RBC, which is red blood cell. Now, 34% of this RBC is comprising of hemoglobin. So, hemoglobin. So, globin is the protein part, hemo is a group that contains iron. So, hemoglobin is a iron containing pigment, iron containing protein which is present in RBC. Hemoglobin, sometimes it is also written as HAE. So, people also write like hemoglobin, both are same, but we will just try to stick into one particular form, and that is hemoglobin. In short, we will just write HB. So, hemoglobin comprising 34% of the total weight of RBC. This RBC hemoglobin, since we are talking about this protein is made up of amino acid. So, this hemoglobin is a polymer of amino acid, and there are 4 chains. So, there are 4 chains in this hemoglobin, and these chains of hemoglobin are designated as alpha and beta. So, there are 2 alpha chain and 2 beta chain. These 2 alpha and beta chain is combinedly forming a one hemoglobin molecule. So, this hemoglobin is basically an ingredient that is binding to oxygen. So, one hemoglobin molecule can bind maximum to 4 oxygen molecules. So, one hemoglobin can bind to 4 oxygen molecule in theory, and this binding capacity varies with the partial pressure of oxygen and carbon dioxide present in the lung cavity. So, this is in nutshell about the hemoglobin and its binding capacity. If we start looking into the hemoglobin and its percentage in terms of number, so approximately 100 ml of the blood. So, 100 ml of human blood, we can say just blood, is approximately containing 13 gram of hemoglobin. So, 100 ml of blood contains 13 gram of hemoglobin, and 1 gram of hemoglobin can bind to 1.34 ml of oxygen approximately. So, 1 gram of hemoglobin can bind to 1.34 milliliter of oxygen. That means, if we multiply 13, and we will get an idea that approximately 20 ml of oxygen is what is present in 100 ml of blood. 100 ml of blood. So, this 20 ml is the actual carrying capacity of 100 ml of the blood. But if we start looking into the actual exchange of gases, this is not 20 ml. The actual gas exchange takes place in the human alveolar cavity is 5 ml. So, 5 ml is approximate volume of oxygen that is getting exchanged per 100 ml of human blood. So, this is in terms of oxygen exchange capacity when we are talking about hemoglobin. Now, the hemoglobin dissociation is hemoglobin binding with the blood is very important parameter. Now, if we can draw as depicted in this slide, if we draw the percentage of, so we will have partial pressure of oxygen in this axis, we call it x axis, and in y axis we have percent hemoglobin saturation. So, the hemoglobin molecule is getting saturated with oxygen with different concentration. Now, this graph against partial pressure of oxygen and the percent hemoglobin saturation roughly gives a sigmoid graph, where the 100% saturation comes with 100% of partial pressure of O2. Now, this partial pressure of O2 is very dominating when we have very high saturation point, and in CO2, when the CO2 concentration is very high, or the CO2 partial pressure of CO2 is very high, at that time the percent saturation of hemoglobin is less. So, these two parameters, so here we can nicely say that oxygen is quite partial pressure of oxygen is very high, and here the partial pressure of oxygen is not that high. So, the CO2 concentration is little high. So, the major transportation of oxygen is happening in lung cavity with the help of hemoglobin. So, hemoglobin is the actual molecule that is transported the gaseous oxygen from alveolar cavity to the tissue space. So, this is another this is very important in terms of understanding the transportation of oxygen. Now, important thing about whether this graph is constant is not the case. Normally, human blood is approximately 7.4 pH. If we slightly get acidic nature of the blood, that means if the H plus ion concentration is getting high in the blood, so this graph is getting tilted little bit towards the right side. So, this graph is tilting towards right. If the concentration of H plus ion is increasing and causing this pH of the blood decrease to 0.2 level, that means 7.2. So, instead of 7.4, if the blood is showing a pH of 7.2, then what is happening, the whole graph, oxygen dissociation graph. So, this graph is called oxygen dissociation graph, oxygen dissociation graph. So, this oxygen dissociation graph is shifting towards the right side, and opposite is also true when we are getting an increase in the pH. So, if the pH is getting increased about 7.6, the whole graph is shifting towards the left side. So, this shifting of this graph towards the left side is because of increase in the pH of the blood, 7.6. So, this shifting is depending upon the H plus ion concentration, and this phenomena is what is very important in order to understand the actual oxygen carrying capacity in the lung. So, there is another parameter which is very important, and that is carbon dioxide. So, if carbon dioxide concentration is also increasing, then this graph is also shifting towards the right side. That means, if the carbon dioxide concentration is high, the oxygen binding capacity is low, right. So, in that case, the graph is shifting towards the right side. And another important parameter is temperature. So, temperature also plays a very important role. So, if the temperature is very high, then the whole graph is shifted towards the right side. So, this right shifting phenomena is very common when we have high concentration of H plus, that means acidic pH, if we have more CO2 concentration and more temperature. These three conditions are quite common at the level of tissue capillaries. So, in tissue capillaries, the tissue where the exchange of gases taking place, and at that level the H plus CO2 and temperature is very high, and because of this region the whole graph is shifting towards right side. That means, so this is what is important in understanding this oxygen dissociation curve. So, next comes the second important ingredient which is required to transport oxygen, and that important ingredient is plasma. So, which comprises of only 3% of the total oxygen transport. So, 97% is governed by hemoglobin and 3% by plasma. Now, we need to understand what is plasma. So, if we take blood in a simple tube, say for example, this is a tube and we are taking blood in this tube. So, if we take as such blood, the whole blood will coagulate, right. Now, what is happening if we take the blood in a tube, the whole blood will coagulate, and when the blood coagulates, it will give a little bit of slightly straw color fluid in the top. So, we will get a small volume of straw color fluid in the top once the blood get coagulated. That fluid which is coming on the top of that clotted blood is called serum. Now, this clotting is an inbuilt phenomena in blood. So, blood can clot anytime if it exposed to the atmosphere. If anybody is adding any chelating agent, agent stands for the one which can take out calcium from the blood, and one such chelating agent is EDTA, ethylene diamine tetraacetic acid. So, this is a chemical agent which can take out the calcium from the blood. So, if calcium is taken out from the blood will not coagulate. So, if the blood will not coagulate, we will not get serum. So, instead of serum, so if we add little bit of if we repeat the same experiment, and in this experiment we will take little bit of this chemical called ethylene diamine tetraacetic acid, and if you collect the blood in this tube, if we mix it, then after some time the RBC will settle down, the red blood cell will settle down, and we will get a clear cut area of the fluid. So, we will have the cell mass, so we will have cell, and then we will have little bit of the fluid. Now, this fluid and cell part will be segregated out when the blood is not clotting. So, this fluid part of non-clotted blood is called plasma. So, this plasma is what is carrying 3% of the oxygen, remaining oxygen from the blood. So, these two, plasma and hemoglobin, are important ingredient for transportation of oxygen from the gases. Transport of gases, we will discuss about the CO2 transportation. So, we discuss about the O2, and now we will discuss about the CO2 transportation. So, CO2 transportation is quite different as compared to O2 transportation. So, in O2 transportation, we discussed that approximately 5 ml of oxygen is getting transported per 100 ml of the blood, while in case of CO2, only 4 ml of CO2 is getting transported per 100 ml of the blood. The CO2 transportation is quite different as compared to O2 transportation in terms of exchange. So, CO2 is getting exchanged in the tissue level. So, we need to understand what is happening in the tissue level in terms of the exchange and all. And if we start looking and see what is happening in the tissue level where the CO2 transportation is taking place. So, we have the cell here. So, this is a cell. So, we will just write tissue, and here we have capillary. So, we will just write capillary. So, capillary. So, these are blood capillaries, and these blood capillaries are present in the tissue area. Now, the partial pressure of carbon dioxide is very high in this area, and that is why it is getting transported across the tissue capillary. Now, this transportation is happening in two forms. Either this CO2 is getting directly into the area which is the fluid in nature, right. So, this we can call the plasma. So, we have this fluid in the form of plasma. So, the plasma is the one which is carrying the CO2, and this is approximately about 7% of the total volume of CO2 transportation. So, plasma comprising only of 7% of the total volume. Now, next comes the rest and the major amount of carbon dioxide carrier, and this carbon dioxide carrier we need to understand about RBC. Again, the same hemoglobin is playing very important role in transportation of CO2 also. Similarly, like what we understand, the transportation of oxygen is carrying mostly by hemoglobin. Here also the hemoglobin plays a very important role in transportation of CO2 also. Now, this is a single cell of RBC, single red blood cell in the capillary. I am just drawing it in order to give you an idea that what is happening here. So, this carbon dioxide is combined with water and forming a molecule or carbonic acid H2CO3. CO2 combined with water to form H2CO3, which is a carbonic acid. Now, this conversion is very slow in normal process, but in RBC we have an enzyme which is called carbonic anhydrase. So, the enzyme present in the RBC is called carbonic anhydrase. So, carbonic anhydrase is an enzyme that can facilitate the rapid conversion of carbonic acid production from CO2. Now, CO2 combined with water to form carbonic acid. This carbonic acid is again dissociating into H plus and HCO3 minus ions. So, I will just draw it little bigger in order to give an idea. So, this is just a bigger form of RBC. So, what is happening? This HCO3 is getting dissociated H plus and HCO3 minus ion. So, this bicarbonate ion HCO3 is called bicarbonate ion. So, this bicarbonate is comprises of 70% of the actual or the total transportation of CO2 from the blood. So, this bicarbonate ion is the major ingredient which is transmitting CO2 from this blood capillary. Remaining H plus is very important ingredient because this can maintain the acid base buffer equilibrium inside RBC. So, this H plus is binding to hemoglobin to form hemoglobin molecule and maintaining the acid base equilibrium inside RBC. Now, what is happening here in terms of ions? Bicarbonate ion is getting out. So, to compensate this loss, the RBC is taking some chloride ion from the fluid. So, chloride ion, so this is Cl, chloride ion is getting inside the RBC. Sometime this phenomena is called chloride shift. Now, this chloride shift, as such we are not going to discuss this one in much detail, but just to maintain the equilibrium something needs to go inside RBC in order to maintain the ionic balance between the outside and the inside environment in blood. So, just to compensate the loss of bicarbonate ion, the chloride is moving in and the phenomena is called chloride shift. So, 70% by bicarbonate, 7% by the dissolved plasma volume of CO2. Now, rest 23% is coming from a very important ingredient which is formed by the amine group present in the hemoglobin. So, the amine group present in the hemoglobin is bind to the CO2. So, this hemoglobin and the CO2 after binding can form a very specific molecule called carbaminohemoglobin. So, carbaminohemoglobin is comprises of 23% of the remaining CO2 exchange from the blood. So, CO2 is totally transmitted in three forms: plasma 7%, bicarbonate 70%, and CO2 dissolved, CO2 in combination with hemoglobin in the form of carbaminohemoglobin, approximately 23%. So, this transportation is very important, and this transportation of carbon dioxide in the carbaminohemoglobin and the plasma and the bicarbonate is maintained by the partial pressure, and it's fluctuated or it's get modulated by the variation in the partial pressure of carbon dioxide. Next subheading is regulation of respiration. Now, this regulation of respiration is very important in order to understand how this respiratory process is getting controlled. So, this whole inspiration, expiration, the breathing phenomena, the deep breathing, the shallow breathing, these all breathing phenomena and the inspired and expired volume of air in and out from the lung cavity is very important, and this all exchange of gases and everything is controlled by our brain. So, to understand this regulation, so we will understand this regulation of respiration. Now, this regulation of respiration is governed by our central nervous system, which is comprised of brain and spinal cord. Now, the major part that is responsible for maintaining this regulatory aspect of respiration is brain stem. Now, this brain stem is a specific area in the brain located in the downside of the central nervous system. So, this slide is just summarizing some of the important structure in the brain stem that is regulating the respiration. So, this whole brain stem is divided into many such parts, two aspects. So, one is called medulla and the other is called pons. So, this medulla, sometimes it is also called as medulla oblongata, and the other one is pons. So, medulla oblongata is divided into two as shown in the slide: dorsal respiratory group neurons and the ventral respiratory group neurons. So, the dorsal respiratory, so these two are the part. So, one is dorsal and the other one is ventral. So, dorsal and ventral, these are the two group of neurons present in the medulla oblongata that is maintaining inspiration and expiration respectively. So, the dorsal can regulate the inspiratory movement of the air, and ventral group of neurons can regulate the expiratory volume. So, expiration is basically controlled by ventral and the dorsal is for inspiration. Now, pons is what is very important, and this is called Pneumotaxic Center. So, this pons as shown in the slide is called Pneumotaxic Center. So, actually rate and the depth of breathing is controlled by pons. So, pons is controlling rate, the maximum breathing rate and the depth of respiration, the inspiratory and expiratory reserve volume and the other volumes which we discussed in the past classes is basically controlled by pons, and inspiration and expiration in specifically controlled by medulla oblongata. So, this is the regulatory aspect of this respiration where we need to understand these two very specific structure of the brain stem. Now, the brain stem, this two structure is located in central nervous system, which is in the top of our body area. Now, our lung is here and the brain is here. There is a connectivity in between this brain and the lung cavity, and which is normally called as blood brain barrier. So, this blood-brain barrier is very important structure that can separate the brain from rest of the body. So, H plus ion and CO2 concentration is the major ingredient. So, what we discussed throughout our lectures or throughout our this topic of respiration, that we have oxygen, we have carbon dioxide, and we have H plus, and sometimes we have temperature. So, these four are the players that can maintain or regulate the respiration and all. Now, O2, oxygen, is not that important in terms of regulatory aspect. So, oxygen is directly controlled from the carotid artery as such. So, carotid artery and aortic arch are the two areas that can control oxygen as such. So, oxygen is not that deciding factor. The actual deciding factor is H plus and CO2 ion. Now, CO2 and H plus ions and CO2. Now, H plus ion is not crossing the blood-brain barrier. So, H plus is again not taking actively participation in regulating this phenomena, because these are unable to cross the blood-brain barrier. So, this is having again limitation. Temperatures is not playing any role in terms of blood-brain barrier, but then CO2 is very important. CO2 can diffuse nicely from the blood-brain barrier from lung to the brain area or central nervous system. So, CO2 is kind of activating the area with the different aspect, and because of this, the CO2 concentration in the atmosphere is mainly regulating the actual aspect of respiratory system. These are some of the regulatory aspect, and now we discussed that we have this lung, and if we take a cross section, so cross section means cutting the lung in either vertical or transverse. So, this is called vertical and this is called transverse. So, any cut can give the actual picture. So, if you take a cut and if you start looking into the microscope, we can see the nice alveoli pattern. So, these are the nice alveolar pattern. Inside the lung. Now, sometimes what is happening, because of some external infection, either in the form of a bacteria or in the form of a fungal infection, the fluid which are present in between this area is getting internalized. So, the fluid is coming in this area, this alveolar area, and in addition to fluid, we are getting some RBC and WBC in this area. That means, what is happening, because of the problem, because of the bacterial infection, because of the external environmental fluctuation, because of certain kind of fungal infection, we are getting fluid accumulated inside the alveolar cavity, and in addition to fluid, what we are getting is RBC and WBCs also. Now, accumulation of fluid, RBC and WBC is not good, because that can obstruct the passage. So, this area is for air. Now, instead of air, we are getting this kind of unwanted material. So, the volume of the actual lung cavity is not that much. So, this condition where we are getting this inflammatory cells, and sometimes it is called inflammatory cells. So, these inflammatory cells are getting accumulated. So, this kind of instances in lung is collectively called as pneumonia. Anything related to pneumo stands for air. So, anything related to air is called pneumonia, as we discussed in our poems, that we have Pneumotaxic Center. So, Pneumotaxic Center is something that can maintain the pneumo. Pneumo means air. Sometimes we have an extra air inside the air, this cavity, the chest cavity. So, we also called it pneumothorax. So, this is one such phenomena. Now, there is another condition. Sometimes in cigarette smokers, when the person is constant or chain smoker, what is happening, the irritation which is coming from those cigarette smoke is not good for lung health. So, this cigarette smoke is causing constant irritation in this area, because this is the last one. This is the last one which are getting affected. When the air is sucking from the nostril or from the air passage or from the mouth, this is coming after passaging through all those tracts, and this is the last reservoir area, alveoli. Now, because of this constant irritation, sometimes this whole interconnecting membrane is getting dissolved, and because of this dissolution, the whole alveolar air area is getting squeezed into a single big chamber. So, as such, the whole alveoli surface area is getting decreased because of this fusion of the membrane, and this fusion of the membrane, because of this constant irritation coming from the outside cigarette smoke or any kind of other external element, is called emphysema. Emphysema. So, these are the condition which is not good for lung health. So, some of the condition which we need to understand is emphysema and pneumonia. So, two other conditions which we need to understand apart from pneumonia and emphysema is asthma. Asthma. So, this asthma is we are not going to go and talk much about in detail of this, the detail of this asthma, but just to get an understanding of the terminology, this asthma is kind of inflammation. Now, inflammation is a normal terminology people used to kind of use in terms of giving any deviation from the normal process. So, inflammation means any deviation from the normal condition of bronchi and bronchioles. So, bronchi and bronchiole if getting inflamed, that means those inflammatory cells like RBC and WBC which are getting infiltrated inside the alveolar cavity. Instead of alveoli, here we are talking about bronchi and bronchioles and some fluid coming from outside, the condition is called asthma. There is another condition where the disease is associated with the occupation. So, those are called as occupational respiratory disorders. So, occupational respiratory disorders. Now, this occupational respiratory disorders are the disorders which are related to respiratory system, but are mainly in context to their occupation. So, like the person working on the grind mills where they are thrusting the stone and all. So, there is a constant influx of the dust particle and also the person which are involved in road construction and all, the constant coal tar absorption from the atmosphere. Also somebody involved in asbestos factory. So, those kind of ingredients which are constantly coming inside the lung cavity can cause inflammation, cause irritation, and those irritation and inflammation can lead to accumulation of fluids as well as the inflammatory cells which are in the form of WBC and RBC inside the respiratory system. And because of those disorders, the person can constantly cause having a breathing problem, sometimes having a cough, sometimes having constant kind of sneezing and all kind of respiratory distress, and those are all related to occupational respiratory, those coming under occupational respiratory disorders. So, in total we understood some of the aspects related to this respiratory disorders. So, either there is a pneumonia, either there is a constant irritation where the whole respiratory membrane is getting infused and forming a big cavity, and the condition is called emphysema. Either there is a bronchi or bronchioles inflammation, asthma, or related to occupation, which is called occupational respiratory disorder. So, in total we discussed about the composition of air, why this air is not exchanged apart from oxygen and carbon dioxide. So, why only oxygen and carbon dioxide is carrying, not nitrogen and other elements which are present in large quantity in atmosphere. So, there are certain regions which we discussed in the first lecture. Then the total anatomical and physiological aspect of our respiratory system, how the respiratory passage, the whole chest cavity, whole thoracic cavity is bound nicely with the rib and the intercostal muscle, and how the respiratory system is controlled by inspiration and expiration, and with the help of diaphragm and the muscle present in the chest cavity. And later on we try to look into how the gases are exchanged from the lung cavity and from the tissue capillaries in order to exchange the oxygen and carbon dioxide from the atmosphere and in the lung cavity. So, this is in a nutshell, and about this whole topic of breathing and exchange of gases.